Hi, my name is Jordan Moberg Parker, and I'm the Director of Undergrad Research Laboratories in the Microbiology, Immunology, and Molecular Genetics Department at UCLA. And today I'm going to be discussing a method for helping students learn how to read the primary literature. This is an adaptation of a workshop that I've given to instructors and postdocs and grad students who are interested in helping students read primary literature. So all the materials I'm referencing today can be found in this shared folder. So you can see the handouts and slides and other things there. And I'm also going to be sharing with you a website that we've created to also help with the grades process. So the grades process was originally developed by Sally Hoskins in the City College of New York, and I've made some adaptations and prepared some learning materials to help students and instructors tackle this process. So we're going to start out with a little bit of background on the process, and then we're actually going to use an example paper to actually work through the process. And so you'll be able to use those detailed creates instructions, the worksheets and articles, summaries, whatnot, that are posted into the box folder to follow along if you like. Okay. So let's get started. So our learning outcomes for today are to basically kind of understand what the whole approach is and appreciate the value of incorporating primary literature into your teaching. And so by the end of this, you should be able to know what the process of the steps are of the process and apply them to an article and figure out which steps that might be most appropriate for you to use in your own classrooms. All right, so primary literature. What are the benefits and the barriers for using primary literature with students? So some of the barriers, let's start with, is that students often find these to be extremely frustrating, confusing, time-consuming to read, challenging, overwhelming. Fortunately, they do find it interesting as well. But it seems to be kind of a process, and especially the idea that there's different kinds of vocabulary that are used in articles can be really difficult for students to dive in. But the pros are that using the primary literature and reading the primary literature can help enhance students' critical thinking skills, their quantitative literacy, and help them really learn how to start to analyze data and to develop scientific arguments. But again, this can be a struggle for students. The format is weird. It doesn't actually follow a narrative about what necessarily happened in order. Terminology can be difficult, and the reasoning can just sometimes be overwhelming. And for me, one of the big things about scientific papers is that there's not really a narrative flow. So here are all the parts of the paper that we're all familiar with. Title and abstract, of course, introduction, and then all of the methods are grouped together, and then a series of results, and then all of those things have to be kind of combined together and discussed in a discussion. But this doesn't necessarily reflect the scientific process about iterations of experiments or how one experiment really leads into the next. And one thing I also noticed in my experience working with students is that they struggled with reading these papers. In particular, one of the things that we do in my curriculum is to have students present an article in a journal club format. And it's really, I really saw that these students struggled to present these. So these were actually pretty advanced students. They were 3rd and 4th year upper division students doing research in our course-based undergrad research experience. And for this process, I was spending hours coaching individual students in how to do this. And the end products were still not great. The level of the analysis, the quality of the analysis, and the narrative that they were telling as they were giving their presentations was all really surface level. So I was actually at a science education conference and heard about this process called CREATES. It's a structured process for analyzing the primary literature. One of the things that makes this process great is scaffolding. So basically guiding through students in a step-by-step way in which you first have them build foundations and then introduce incremental steps and build upon their knowledge. And so the CREATE approach is a way to do this in the primary literature, with the overall goal being for students to work through the data of a paper as if they had generated it themselves. And so basically, they need to learn how to do each of the steps and then take all that and synthesize it into a cohesive narrative. All right, so what is the CREATES approach? So CREATES is an acronym, and the various parts of it are, the first one is C, concept map, the paper introduction. Then R is read, so read and annotate the results. And this also includes diagramming or cartooning out the method sections. And I'll show you some examples of what I mean by this. The next is E, elucidate the hypotheses for each experiment and really figuring out which of those experiments are going to be addressing the hypotheses. And then A, analyze and interpret the results section, relating those back to the hypotheses. And then the next is TE, think of the next experiment. So what are the unanswered questions that still need to be addressed? And then finally, synthesizing all of these parts into a cohesive narrative flow. So basically making a concept map of the whole paper. Okay. So there's quite a few steps to this. So this is why it needs to be scaffolded. You can't just throw this at the students and say, go for it. You have to teach them each of the parts. And which parts you focus on is going to vary depending upon what your own learning objectives are for your students. So which steps you emphasize are going to vary. So are you using this as a supplement to lecture or lab to get content? Are you trying to teach them science process skills? Are you just, or are you trying to get students to do a deep dive into the literature for something like researching their own project, or to give something like we did, like a journal club presentation? And so once you know kind of what all of these steps are, you can figure out which are going to be the ones that are most important for you and for your students. One difficulty for implementation really is kind of the learning curve of the process. So that's why the scaffolding is particularly important. And to help with this, I worked with a group called WIRE, the Writing Instruction and Research Education group at the UCLA Library, to put together a set of detailed examples and templates that you can use. So in addition to watching this video, I highly recommend that you check out the CREATES website that we've worked on. So again, have lots of examples and templates and step-by-step instructions. All right, so in the meantime, let's actually go through and talk about each of these steps. So how do you go about scaffolding the CREATES process? So we'll start with C, concept mapping, the introduction. The first part of the scaffolding is, first of all, to make sure that your students know how to do concept mapping. And if you're not familiar, concept mapping is a way to basically visualize the concepts and make diagrammatical linkages between the different concepts, specifically saying how the different concepts link together. Okay. So you usually start out with a kind of key concept, and then you have linking phrases and talking about the relationships between the nodes. And the idea is to basically identify what the whole big point of the paper is and how the different parts of it fit together. So I recommend starting with actually just teaching students concept mapping. And I like this because it's very meta, but here are two concept maps on concept maps. And so very meta, the concept of concept mapping. So you can see that there are, each of the concepts are labeled and there are linking words, and there can be multiple kinds of hierarchies and flows in these concept maps. One important thing that I talk to students about when we're doing this is that there's no one right way to do it. So we'll usually start out with a practice concept mapping activity. So for this activity and for the subsequent article that we're going to be discussing, I'm a microbiologist. So we're going to be reading or discussing a paper that's microbiology related. And something that most life science students have all experienced at some point is genetics in some form or another. So for an activity, I would have students do a practice concept map for genetics. So here are some keywords that I would provide to the students and let them know that they can add any words that they would like. And so I'll have the students work individually for a few moments and use these words and prepare their own concept map, making sure that the key ideas are linked by words and phrases that explain the connection between the ideas. So if you're going to follow along for practice, you can pause the video now and see what you come up with for your own concept map. If you're just going to follow along with me, then that's totally fine too. The next thing I usually do at this stage would be to then have students get together in pairs or small groups and compare their concept maps. And here the students can see that there's different ways to connect the ideas and that everyone's concept maps are different. And then we will often have the students revise their concept maps and try and put together kind of a master map. If you have plenty of time in class, you can then have students go up to a whiteboard or blackboard and draw up some examples, and then you can point out interesting connections that the students have made. All right, so once we've established that concept mapping is something that is in their repertoire, then we can move on to an actual paper. So if you're going to follow along with an example, I've prepared materials for this particular article. So if you want to follow along and do the steps with us, then this is the article that we're going to be using. So this article, lineage overwhelms experimental conditions in determining rhizosphere bacterial community structure in a cosmopolitan invasive plant. So what does that mean? I'll give some general background in a few moments, but basically that there's this group of plants, and some of these they're all different, or all the same species, but they have different lineages. They are the same species, but they originated in different parts of the world. And when you grow them together, they have variations in how they grow. So if this seems like an overwhelming topic, a concept that you're not familiar with, or you're introducing something new to students, then it can be a good idea to also find an article that has had like science communication articles written about it. So finding articles written for a general audience can really help novices dive in. Right? So oftentimes these articles written for a general audience are much more explicit about the big picture, and there's not the obscuring terminology that's used in the article. So for students, this is what I might start out with. I'd give them these, there's actually 2 articles on this particular paper, and have them read first, and then I would have them dive in and start reading the introduction. So again, if you would like to follow along and you want to take a quick break, links to the general audience articles are in that box folder that I've mentioned before. Okay. So you should also think about the papers that you're assigning in general. So shorter is not always better. The longest papers are not always better. It's really important to look through the papers that you're going to be assigning to the students, and probably even going through the whole CRATES process yourself. Because with shorter papers, there's usually not enough detailed methods for students to get all the context, right? And longer papers may have lots and lots of different experiments that are done, and you might not want the students to focus on each and every one of those experiments. So it's important that you spend some time and find papers that work for you. Okay. Another option for looking for articles, there's this resource through AAAS, and these are all articles that have been annotated and have accompanying teaching materials. So kind of know that these are good quality, and they can be used in particular texts for your teaching. And they have a whole bunch of different kinds of topics. So if you haven't seen this resource, check that out, Science in the Classroom. All right, let's circle back to concept mapping. So what the students would have been doing here in concept mapping, the introduction, we'd have the students read those articles, the science news articles, and the introduction of the article itself, and start to put together an idea about what all of it means. For a lot of instances, you might need to give some short background information. So you might need to do a background lecture before the students can really dive further into this. So I would usually do this after I'd have the students read the introduction and start kind of identifying the key parts of it. So in this case, I would maybe highlight some of the things about the research problems and questions in the paper, things to look at. So in this case, they're looking at rhizosphere soil. And that's the layer of soil that's closest to plant roots. And it often has a thriving microbial community. But it's also important to know that there's different environmental factors that can structure those communities. And then talk about maybe what are some of the big picture things that the field is interested in. And then starting to help the students kind of pull out some of the research questions for this particular paper. This would be a great time to kind of have a class discussion about this and have students trying to identify, come up with their own ideas about what the research questions are before sending them back to do their concept mapping. For some context about this paper, it's about Phragmites australis, and this is globally distributed, but is also in some places a harmful invasive. So there's 3 lineages of this plant, and they can all grow in the same geographic areas, but they have different haplotypes of the same species. Okay. So they're frequently encountered next to each other, and the introduced is an invasive species that takes over the native habitat. Okay, so background established. Time to now have the students go back and work on their own concept map. All right, so some ways you can break this down for the students is to first give them some ideas about how to get started. And how to get started might be to give them some examples of how to annotate introductions or annotate research articles. So the whole concept map of the introduction has 2 kind of steps. Annotating the introduction, this includes defining all of the terms, identifying the key background information, identifying the research problems or questions, what are the important variables that are going to be in the study, and then once that is done, then actually doing the concept mapping. So here is an example from the website, the CRATES website that we made, showing an actual example of a student annotated paper. So you can see here that the students are identifying questions that they have. What are the kinds of things that are unknown? Here's the big research question they identify. What are some of the key things that they might need to look into a little bit further before they can really understand the paper. All right, and then after that, once they kind of have pulled out all these ideas, then it's time to reconstruct them themselves. So to synthesize all of that into their own concept maps of the introduction. So here are two concept maps from that plant microbe interaction paper. That's our example throughout this presentation. And then just another example from a different article, again from our CREATES website. So you can see here that there are lots of different ways to do the concept mapping, but it is important that the students have these linking words, right? And if you're going to be having the students present these papers, then this is a good way to get started, have students explain the context of the paper using their concept mapping. All right, so let's talk about the next step, the R, which is to read and cartoon the methods. All right, so in this case, the method section is going to be used to work backwards to how the results were obtained. And what we ask the students to actually do is diagram the cartoons and methods. And this doesn't mean flowcharts. I mean actually drawing pictures and cartoons. But the balance of this is it doesn't have to be super detailed. I ask the students to do this in a way that's conceptual. So what's the big picture of the methodology that they're doing rather than procedural? So basically don't get bogged down in the details. They don't need to draw out and include every volume and concentrations, but they should be looking to see what the controls and comparisons are in the experiments that they're reading about. Right. So here's an example of a very simplified cartoon that I've made for this paper we're reading. So here's the rhizosphere soil being collected from the plants and put into tubes. Right? So that's all I would want for that first part of the methods. But the reason why I really focus on this and focus on the cartooning of the methods is that this is a section that many students skip, and often with good reason. There's often way too much information there than we need, but by doing some of this cartooning, then the students can actually put into, they can conceptualize these methods, and that helps put the results into context. All right, so for our example paper, there was kind of two key methods, two big experiments that they did. They did a field experiment where they collected samples of this plant from across North America, and they did a garden experiment where they took common soil and then these different lineages of the plants. All right. So in this case, we'd start to have the students start to think about identifying the questions that the experiments might be trying to answer. So just based on the methods, you can see that you can pull up the idea that there are differences in the microbial communities in these different lineages that these authors might be interested in. All right, so these samples were all collected, and then here's some additional methodologies. So if our question is what bacteria is associated with different lineages, you might have example of the plant, collecting the soil, extracting the nucleic acid from the soil to get the microbial community and doing data analysis with that. Another example, here's how I've drawn out the greenhouse experiment. So taking all three of the different species, the different lineages of plants, removing all of the microbes from their roots and planting them into soil. Soil has not been sterilized, the soil that would have the same bacteria in every pot. And then you put in the plant and see how that might change the bacterial population from where it started. Right, so those are some kind of big picture methods. If you're working on a paper as a class, again, this would be a time when you might also need to do some mini lectures. For this article, there's some pretty complex bioinformatics that are done. So I'm not going to go into this particular tool, but for example, this is a computational tool that will look at quantifying different microbial populations. So I wouldn't want students to try and draw a computer or draw out all of the steps of this analysis, but they should be able to put it into some context with the other experiments. And if there's going to be some figures coming up that graph out the data in ways that the students might not be familiar with, this might be a time to also do some things like discussing some of the analyses that are showing up and being discussed in the methods section. So for this paper, I would spend some time talking to students about principal coordinate analyses. And again, this is quite a complex set of analyses, but in this case, for the students, I would say the only thing you need to know about these is that if there are samples that are grouped together, then they're more similar to each other, right? So context and conceptualizing rather than trying to understand what all of the details are. But again, this is going to vary depending upon your student population and what your learning objectives are. If your learning objectives include knowing the details of principal component analysis, then you'd go into more detail here. All right. Once the methods have been cartooned and sufficiently explained to the students, then step 2B of this is to go now into the results section of the paper. And now that they have an idea about what methods have been done, reading the results to identify which methods led to which results, right? So we're using the methods section to work backwards to how these results were obtained. All right, so again, a couple sub-steps to this. We want to annotate figures and write notes directly on the results section. Right, so again, here is an example of this article annotated and to identify what's happening in the results sections. Okay, so when we're talking to students about annotating, we say highlighters are okay, but every time you highlight something, you need to write a note next to that highlight so you know why you highlighted it, right? Because students can get very highlight happy and just end up highlighting a whole paragraph, but not really remember why that particular paragraph was important. Okay. And then going through the actual figures and adding labels and identifying what the key results look like they are going to be, right? So writing in notes along with the highlighting and annotating the figures. All right, so again, this is the article that we're using for our example, and we can see that the different lineages of the plant are in these different colors, and each dot represents an entire microbial community, and you can see that the various communities, those dots are clustered closer together, meaning that no matter where the actual plants came from, their bacterial communities were similar if they had the similar haplotype. And to show some additional examples of this, so here's an example of some additional student work. Here's the methods that have been cartooned out. You can see that this is a percent survival graph. We've got our live mice over course at dead mice to look at survival curves. Right? And the students have outlined kind of what each is happening in each of these parts of the graph. Okay, so those are just the first two steps. We still have a couple to go. All right, step 3 is elucidate the hypotheses. And so again, some sub-steps to this, starting out with the overall research question. This might have already been identified in a previous step. So maybe the students identified this and they're talking about the introduction, right? But now it's time to kind of put all of these together in one place, right? So you might have found this in the introduction from your concept map, and you need to have some kind of specific details what this specific paper is asking about. Okay. So what's the big picture hypothesis for the whole paper, if there is one? And then the next step is to then, now that you've identified the methods and which results go with those methods, what was the hypothesis that links those things together, right? So in order to do this, you think about what kind of experiment it is, right? So we've kind of just, for simplicity's sake, broken it down into two general types of experiments. Right? So once you've identified all of the experiments in the paper, we ask students to think about, is this a descriptive study, an experimental test, or is it something that they can't quite figure out? It doesn't fit nicely into one of those two boxes. Okay. So things like descriptive studies look at existing conditions, and an experimental test involves actually conducting an experiment where you change variables. So for this paper, that's our example paper, a lot of it is a descriptive study, so looking to see what is there in a particular environment. And if the figures don't fit exactly into any of these, you can actually just say it's other. On the CREATES website with the library, we have actually some examples of these and templates that you're going to use for looking further at these ones you've identified what it is. Okay, so what kind of study it is going to influence potentially how you describe the hypothesis. All right, so when I ask students to elucidate their hypotheses, I have a pretty rigid structure I ask them to use. And I ask them to use since, if, then statements. And of course, not all hypotheses can be forced into this structure, but I have the students try at least. I do this because I think it helps students put, again, everything into context. So the since, if, then statement is a way of formulating them to help them think about why the researchers did the experiment, what's the actual question that they're trying to answer, and what the expected outcome is. So that's the since, if, then. The rationale or the observation that led to the experiment, what the experimental question is that's actually being tested, and then the expected results. Sometimes authors will say in their manuscripts, in their papers, like, we hypothesize that, and students get really excited about that. But then they still have to synthesize this into this structure so that they can think about, again, those three parts: the rationale, the question, and the expected outcome. Right. Sometimes there's not an expected outcome. So this is especially the case for descriptive studies. You know, the expected outcome might be super broad. You know, we want to look to see what the population looked like. And that's fine. All right. If you're going to follow along with us here and you want to pull up that, that paper, now you might think about picking one of the experiments from that paper and seeing if you can put the a hypothesis into that since, if, then framework. All right, so let's see an example for this particular paper. So here's the rationale: since soil and microbes are typically controlled by local soil conditions. So this is a previous observation in the field of microbiology that local environment drives microbial communities, right? So since this is a previous observation, and usually I would actually have students also include the citation here that shows that. So since this is the previous observation, if the lineage of the plants is forcing the microbial community to be restructured, right? So that's the question here. The soil microbes might be influenced by the environment, but is the plant really changing what microbes are there? So that's the question in the if. All right, if that is true, then what might they expect to observe in these experiments that they've conducted? Right, so here there's a couple potential expected outcomes that go along with this research question. Right, so they might expect to see the bacteria communities be more similar to each other within a particular lineage. And then another expected outcome also, if this is true, is that if you take those three lineages and put them into communities that have the same bacteria, then those communities might change based on the plant that's there. Okay. So again, this is trying to put all of the parts of a particular experiment back together. Okay. Then the next step is to say, okay, is the hypothesis supported or not? And to do this, we have the students kind of go back to what they've done so far and work through a specific set of questions about each figure. Right, so now explicitly relating the findings from the article to these hypotheses. So, and making sure they can see which results address which hypothesis. Right, and also having them think about this. How convincing are the results? Right, and actually coming up and writing for themselves a general list of interpretations and conclusions from the experiments. On our website, we've got actual Google Doc templates for students to do this, so they can make a copy of the template for each experiment and work through each of these questions. These templates ask students to write down what the official title is for that figure table, and then put it into their own words, and then answer some specific questions about comparisons and conclusions that they're drawing based on the results. I find that these analysis templates are really helpful for novice students to help students figure out what they should be looking for when they are analyzing data. Okay. But I found that most students, when they've kind of got the hang of this, stop using the analysis templates in detail. They kind of know what to look for and can kind of put this into their own words in their own notes. All right. So the next step, and this is quite related to the analysis and interpret the results, what are some questions that are still unanswered? So what's the next experiment? And part of this is often for the paper, the results of one experiment lead into the next experiment in the paper, right? So having the students identify which experiments lead to which next experiments, and then to also think about what are the overall questions that are still unanswered. And for that, we have them then propose a next experiment or a future direction. And we also ask them to cartoon or diagram that out like they did for the experiments in the paper. Right? So I want them to still be thinking about what's the specific methodology that's going to be used to do that experiment? What controls you need? What are the actual experimental conditions? So they need to actually think quite specifically about how they would conduct this experiment. And a fun thing that you can do if you have the class time is to turn this activity into a grant panel where we have students who've all read the same paper propose their next experiments, and then the other students can give some critique on those, and then they can vote on the best proposed experiments to be funded for further research. Right. All right, so now altogether, the students have taken this article and exploded it out into all of its various components, right? So they've deconstructed this entire article into each of its component parts. And now what we ask them to do is Humpty Dumpty it back together again, right? So now it's time to synthesize each of those components into a cohesive narrative. And this is the final step of the CRATES process. So I asked them to make an overall concept map of the outlining, the narrative of the paper. And to do this, I usually ask students to get some big paper. When we're doing this in person in classes, I'll provide large format paper for the students to work on. And it's often helpful to have multicolored utensils, different colors of pens, colored pencils, and all of the materials that they have worked with the previous steps. Okay. And then to go back to that idea of concept mapping. What's the overall research question? And writing that down in the center, and then branching out from there. So here's the overall research question. Here's the first hypothesis, and what's going to be coming off of that. Right. So the rest of the concept map will be branching out from the overall research question. Right. Because remember, we have them do the synthesizing step because the parts of the scientific paper don't have a flow. So we want the students to reconstruct that back into something that makes sense to them. And I also tell them that it doesn't matter what the narrative was of the authors. You know, how are you going to put it together? Which experiments do you think are the most important? Which ones do you think make sense together. Right. And so focusing on the narrative flow. Right. As I said, identifying the overall research question, which hypothesis is testing that question, which methods test the hypothesis, the results that came from those methods, the conclusions that were drawn, and then is there a new hypothesis? Do they have to revise their original hypothesis? Or these results lead them to a new question and then starting over again? So here's an example. So this would be the research question in the center and 3 different hypotheses that come out of that with the various steps. Okay. All right, so again, having the students come up with their own flow. And the reason I like this is because it's messy, right? Papers are presented linearly, right? This order of things happened. But in reality, anyone who has worked on a research project usually knows that there's multiple things going on at a time, right? And this experiment doesn't always lead directly into this experiment. You have some offshoots, and you don't necessarily do things in the order that you present them in the paper, right? So this shows that it's not just a clean series of events, but it's actually a process. So I feel like this is much more reflective of what we actually do. All right, so I'll show you some examples of student work to see how this all comes together. So again, so these students could have expanded upon their research question a little better, but you can see that there are hypotheses that are coming out from the central question, and then methods that are testing those hypotheses and the various results that came from them. Right. Another example, this one I believe you can see on our website. So again, lots of nice colorful organizing. The students had quite a few different steps going on here. So like I said, there's lots of different ways. And again, there's no one right or wrong way to put all this together. All right, so that's a lot. What do the students really think about this process? Spoiler alert, yeah, it's a lot of work, right? And when we ask the students reflections about this process, they often think that it can be tedious, but they think it's really important to help them understand, to thoroughly read it, deeper understanding, basically better research questions, and breaking out all of the parts seems to be really useful. All right, so it is a process, and your students may want your students to focus on different aspects of it. So this is the kind of thing in which students don't necessarily like it, but they can see how useful it is. I've had a lot of students come back to me over the years and say that, hey, I learned this process and now I'm in these other upper division classes and I have to read papers all the time and I use parts of the CRATES process in these other classes. You think about it, many of us weren't taught specifically how to read articles. We just kind of learned as we went. So having a framework is good. All right, so is this whole process worth it, right? This is a lot of work to go through. So I've done some assessments with students, and this is just a little bit, a snapshot of some assessments that I've done. So I had students giving journal club presentations, as I mentioned at the beginning of the video. So I had cohorts of students, some cohorts that I was individually coaching the students how to read papers. Granted, this is of classes of 30 or more students, so you can imagine how much work that was. Right. Then I had cohorts of students that I implemented this scaffolded CRATES process. And this meant I was doing much less work because this became a student-centered process. Rather than me interpreting the papers for them, the students were doing it themselves, and I was giving guidance when necessary. Okay. So what I did is I used rubrics to assess the journal clubs for the 2 different cohorts, my control group and my CRATES intervention group. And I had a couple different learning objectives involved, and also I had different rubric items identified by higher order and lower order cognitive skills. And I found that some things I was really interested in actually did improve. Students were much better able to identify and elucidate and describe the context of hypotheses. They much better understood the methodologies of the papers. And for me, this was a really important one. They had a better narrative flow. So they could tell a story with the research. And to me, that was valuable. Because let me tell you, I sat through a lot of bad scientific presentations. And if we can help the next scientists tell better stories with their research, that will benefit everyone. All right. And overall, I found that they did have overall higher order cognitive skills were improved in the CRADES cohort. Okay. We also asked the students which steps that they used the most or found the most helpful. And the reading, which is the cartooning out the methods and annotating the figures, was helpful. Elucidating the hypotheses were helpful, and the final narrative concept map was also very helpful. Also notice that students don't always know what's good for them, so I take these results with a little bit of grain of salt. All right, so that is the big picture of the CRADES process and a description of kind of how I've used it in some of my courses. Again, if you're interested in doing this, because a big hurdle for implementing this process is learning the steps and having examples about what you should do, we have this website that you can look at that is full of student examples. And again, this was created by a group at the library with instructional materials from me. So with that, I'd like to thank that group, the WIRE group, especially Doug and Keon and Alex, who really worked on that website. My other instructors in MIMG that have also implemented these programs, and some funding that went into assessing the overall process. All right, thank you, and I'm happy to answer any questions that you may come up with about implementing the CRADES process in your own classrooms. All right, thank you so much, and have a great rest of your day.